Good for you guys. So protein structure. We call a polypeptide a very long chain of amino acids. So when you were doing your models, making those models, I'll kind of pull it up here. When you started out, before you did all the taping together and so forth, you had a long chain of these colored squares. And that was your polypeptide chain. Okay, it looked like a ribbon. But a protein is not that's not a functional protein yet. It has to go through several stages of structural changes before it becomes functional. And that's what this slide's going to kind of talk about. There are 4 levels of structure to polypeptides. The first is just the primary structure, which is the order or the linear sequence of those amino acids. It's the order in which you put them in. And we just talked about what molecule kind of determines that order. What important molecule? It was just in the homework. RJ, you even gave the answer. What molecule determines the order of amino acids? DNA. The DNA order. Okay, those nucleotides. Okay, so that determines the linear sequence. What happens next is that you start forming these alpha helices and these beta sheets. An alpha helix is like a spiral, and in your proteins you made a spiral. And then a beta sheet is this part right here, kind of that wavy pattern. And those start to form along the chain when certain amino acids interact with each other. Okay, and it's just kind of like, but we're still a long linear sequence, but now we're just taking on more shape. Instead of just being a ribbon, we get these wavy parts and these spirally parts. And then we get to the tertiary structure, and this is, I think, the most important part. I mean, they're all important, but this really gives proteins its function, is that it starts to take on shape because those R groups interact with one another, and the protein starts to fold. We get folding. And that's when you were taping those triangles together, and you got this three-dimensional looking shape. Okay, and that's when the protein folds. And that shape is important because that shape dictates the protein's function. Oh, come on. Internet. Now, what we didn't get to, though, was the last part of function, or we didn't make it in our activity, was quaternary structure, and that's when you get more than one of these chains interacting. So if I grabbed another one and somehow got them to stick together, I don't know, like that maybe, that would be quaternary structure. Now we have 2 polypeptide chains. Oftentimes they're identical polypeptide chains, just all kind of working together. Other times there are 2 different ones or 3 different ones, depends on the protein. And that's when you get your functional protein. So usually it's not until you get to this quaternary structure that you really gain any function. Okay, so quaternary is when you get your functional protein. It's an A.
So the ones we made weren't functional?
Correct. The ones you made would not have been functional yet because there was just one polypeptide chain. Usually you have more than one interacting. For example, this picture here, this is hemoglobin. Hemoglobin carries oxygen in your blood. Hemoglobin is a 4 polypeptide chain. One here, one here, one here, and one here. And the folding of those chains are very important because the way it folds, you get this central part right here, this heme group, which I'm circling right there, and there's one here, and there's one here, there's one here, and that's where you carry your oxygen. And so the oxygen has to attach right there. And if that folding is not correct, it won't transport oxygen as efficiently. If one of these is not correct, the protein will only carry 3 oxygen molecules or atoms. All right, and it will drastically reduce its effectiveness of carrying oxygen, which reduces its function in the body. Okay. Now, ultimately, it all leads back to this primary sequence, though, because if that order is wrong, it won't helix right, it won't fold right, and it won't interact right with other chains. And so the linear sequence kind of sets up the rest of the structure. Just like if you have a long book, that you have to read. If I start mixing the letters around in all the words that you're supposed to be reading, you just lost all meaning to that entire book. Because the sentences won't make sense if the words don't make sense. The paragraphs won't make sense if the sentences don't make sense. So it's like a domino effect almost. So that's protein structure and how it relates to function. All right, let's do a quick poll. If you want to jump on pollev.com, wolfhbio.edu. Let me pull up the questions real quick here. I have 2 questions I want to ask. Protein structure. Clear results. Actually, I had 3 questions. See what you guys think. What are the building blocks of proteins? This should be easy. This should be easy.
Did you push the button and let us have it?
No.
There we go.
There we go. There we go. All right.
Legos build everything. I know.
That was the analogy I used. I'm glad no one's choosing it. All right. Amino acids. It's okay.
Oh yeah, we'll step on Legos are hurt.
Next question.
My mom said I had to pick up one of those if she stepped on it. Things were going down. Step on you.
What's the name of the bond that holds amino acids together?
Dude, James Bond, come on. No, someone's got to answer James Bond.
James Bond.
Can I answer James Bond? Yes! Someone did it.
I was tempted to put chicken bond up there, but Pepsi bond.
Swag.
All right. What is the correct answer? Correct answer is peptide bond. Okay. Peptide bond. All right.
One more or no?
No, I'm not going to do the next one. All right, protein function. We're going to kind of get through this kind of quickly. I think one of the easiest proteins to understand the function of is enzymes. Because enzymes, they're usually a globular protein. Sometimes enzymes are made of RNA. But it's a lot like the protein we made, where it's just kind of like a glob. It folds in a really weird 3-dimensional shape. It's not long, fibrous proteins like actin would be like in your muscles, where they're long and they work at pulling on each other. They're more of a kind of a blob of protein, blob of amino acids, but it's not just a blob, it's a specific shape. It's a blob. Okay? And they act as biological catalysts. Now, what does that mean? Anybody know what the word catalyze means?
No.
You catalyze something? Okay, yeah. No, not a connector. Yeah, someone Google it real quick.
Yeah, RJ, Google it.
Already did.
A substance that increases the rate of a chemical reaction without itself undergoing any permanent chemical change.
Right, speeds up a reaction. That's my abbreviation for reaction, RxN. Okay? They basically make chemical reactions happen faster. That's what they do. For example, like in your stomach, you have enzymes that help digestion. If you didn't have those enzymes, it would take a long time for your food to break down. You don't have that kind of time. You don't have time to wait for your food to break down and get the energy out of it. You need the energy within hours. Okay? And so you have enzymes that speed up the reactions in your stomach so that food gets broken down faster. Okay? Yeah?
Yeah.
I'm sorry, what?
The RX
Reaction.
Yeah?
Never mind.
Okay. That's just my abbreviation for it.
Yeah.
Okay. Zach?
Enzyme like
We broke
speed
the
up
harm
the reaction,
last
but like not. It doesn't.
Every time I try and make it text.
The enzyme doesn't get used up. Okay, the enzyme acts like a little machine that's like either puts things together or takes things apart, but the machine never really gets used up.
Yeah.
That's how a catalyst kind of works. Good point. Now, enzymes work by what's called the induced fit model, and you can read this and understand it. I'm going to show you guys a video clip that I think is better at describing how an enzyme works. And it's going to have some vocabulary that's a little above our maybe grade level, because this comes from an anatomy book that might be geared towards seniors or freshmen in college. But I think the video itself is good to watch. I'm going to kind of talk over the guy who's talking. I might actually mute the sound if I can figure out how to do that from my iPad.
This is audio button.
Yeah, I know, but we'll see if it works. Okay, it worked. All right. So here's our enzyme. Again, it's a globular protein. It has a specific shape to it. This shape right here is the most important, though. That's called the active site. That's where all the activity is going to take place. These two things are called substrates. It's what the enzyme is going to work on. Okay, it's what the enzyme is going to either combine in this case, or sometimes it separates. But it takes those two substrates, I'm going to pause it there, and notice the substrate fits perfectly in the active site. Now, what if we changed that protein? What if we denatured it? It wouldn't fit anymore. And if it doesn't fit, the enzyme can't do its job. It can't take these two substrates and connect them. Or it sometimes can't take the substrate and break it apart. And so that shape is essential to protein function. And so this active site, we now get this enzyme substrate complex. They bond together. The enzyme changes shape, causing the chemical bonds in the substrates to weaken or change. And in this case, it makes a product. It's going to release that product in a few seconds. But notice the enzyme changed shape slightly. The product is released. The enzyme goes back to its original shape. We now have a product. The enzyme can work on other substrates that fit into its active site. So the enzyme is not used up. The enzyme can work on something else now and make more products. This could be building, I don't know, a protein for us. It could be building a hormone for us. It could be building all kinds of things that your cells need built, and this thing just kind of cranks it out. Questions, Zach?
Oh no, why just my slideshow just reloaded? And it said,
Oh.
it said all my date, the things I edited.
Really, it should have saved.
It said it couldn't save or something.
I'm sorry. Now, these slides kind of talk about just some examples of proteins and how and their functions in our bodies. There's proteins probably have the most diverse of all functional molecules in our body. The most number of different types of functions. For example, we have hormones. Hormones are kind of in charge of your regular cell activities. It kind of helps the cells do their job. For example, insulin tells your cells to take up sugar. Take sugar out of the bloodstream and store it in cells. What do you guys know? There's a certain disease where you don't have, something's wrong with insulin. What's that disease called? Diabetes. Okay, and there's different types of diabetes. It all depends on, some people don't produce insulin. Sometimes people's cells don't respond to insulin. Okay, but it all has to do with sugar uptake. And that hormone, that protein, tells the cell, take in sugar. You also have growth hormone. That one's pretty obvious. It tells your cells to grow. It's how your cells get bigger. Okay? If you don't have enough growth hormone, you're not going to grow very much. In fact, there are certain diseases that you don't produce the right kind of growth hormone or any kind of growth hormone, and people don't grow very much. Okay? That's also a hormone that's often abused by some athletes. Okay, they'll take growth hormone to try to grow their muscles quickly. Because it does involve, it grows cells. Cells get bigger. Your muscle cells get bigger. But there are bad side effects if you have too much of this hormone. And hormones are released by glands, like we have the pituitary and the hypothalamus found in your brain. You have your parathyroid and thyroid gland. You have your pancreas and your adrenal glands, and then reproductive glands, both male and female. Those all release different hormones. Okay, so those are making the proteins that are going to be released into the body to go tell the body cells to do certain things. Like your pancreas is in charge of making insulin. Yeah?
What if they're overly large, if you're born with
Bigger glands?
bigger than usual?
It depends on the gland itself. So sometimes, I don't know if you'd be born with them being bigger, but like I know sometimes the thyroid enlarges because it's seeking out, it's trying to get the iodine that it needs to make its hormones, so it grows bigger to try to find more iodine, I guess. And so it can enlarge and cause problems. But as far as the other ones, I'm not quite certain. Then we have proteins that form cell structures. They basically form parts of cells that give it structure, give it shape. For example, like spindle or cytoskeleton is made up of fibrous proteins that give a framework to a cell. It's kind of like the scaffolding of a house. You've ever seen a house being built? It starts with the studs. That's kind of like the cytoskeleton. It gives the shape of the cell. It also works as a transport network, like highways that kind of run throughout the cell. And then you have these spindle fibers. You guys remember mitosis and meiosis from middle school? Okay, the spindle fibers are what kind of move the chromosomes around in the cell. That's what pulls them apart during cell division. Okay, so that's a structure that provides a specific function in the cell. But you have other structural proteins like, your hair is made out of protein. So are your fingernails. They're made out of protein. Okay, so there's proteins that just provide certain structural components. And then we have cell transport, movement of molecules in and out of the cell. That's going to be a big topic in this unit. For example, this is an aquaporin. It moves water in and out of the cell. It's like a tunnel or a channel. So water molecules can zip in and out of the cell as needed. Okay, so they act like doorways or channels or tunnels for molecules that need to move in or need to move out of the cell. And finally, proteins help fight disease. For example, antibodies. It's probably the most well-known one to many people. Antibodies, I think of them as like handcuffs. If there's like a bacteria or a virus, some kind of thing we call a pathogen, which could cause harm to your body, the antibodies kind of bind to them and kind of glob them up, kind of sequester them so they can't cause any damage, cause any harm. And so your body releases tons of antibodies when you get some kind of infection. It's meant to kind of slow the bacteria and the viruses down, preventing your cells from dying. In fact, they just did an experimental treatment on those 2 American doctors that were infected with Ebola, where they cultured antibodies. Actually, I think they cultured it out of plants. They got plants to produce the antibodies, and they injected those antibodies, like massive amounts of those antibodies, into those infected patients, and the antibodies helped the body clear the virus out, and they're fine. They're healthy now. Okay, so it's a possible. Again, there's more trials need to be done. You can't just say, "Oh, it worked for 2 people; it's gonna work for everybody." They got to look at side effects and stuff. But that's a possible treatment now for a very deadly disease, Ebola. It's the use of antibodies. All right, so that's where we're going to end. I know I have some